Please.
Okay, so we can have interaction among different carcinogens. For example, smoke and asbestos together can cause lung cancer. We can have interaction between the genetics and the environment. For example, individuals born with Papuan mutation and exposed to asbestos.
Bir dakika.
Look, there is somebody who needs to mute their computer, okay? Because I cannot talk if somebody else is talking. So please mute your computer. Thank you. And then there can be gene and gene interaction, which means there can be individuals who have more than one genetic mutations, and these different genetic mutations can interact with each other. This issue I summarize in this article in CA, a Cancer Journal for Clinicians, where I am referring you for more details. We made the cover of this journal. You see here an asbestos fiber in the lung of an asbestos worker. And then here describe the mechanism of the BAP1 and how BAP1 increased susceptibility to asbestos, described in gene for environment interaction. The reason that we got the cover of the journal that has the highest impact factor by far in the world, 3 times that of New England Journal of Medicine, is because gene-environment interaction that we are studying in mesothelioma applies to all cancers. Basically, every cancer in this world is caused by gene-environment interaction. The argument that there is in the scientific debate is whether 2/3 of all cancer are caused by the environmental carcinogens and 1/3 caused by genetics, or whether 2/3 are caused by genetics and 1/3 by the environment. But clearly the two will interact together to give the final result that is cancer. Mesothelioma is a cancer, like all the other cancers. And so mesothelioma can occur because of spontaneous accumulation of genetic mutation. No need for asbestos exposure, no need to be born with a BAP1 mutation, simply bad luck. And here I have 5 examples of mesothelioma that have occurred because of spontaneous mutations. Not in asbestos workers. How do I know that? Well, because this mesothelioma came to me from the zoo of San Diego. The first one is in a lion. The second one is in a cat. The third is in a horse. The fourth is in a dog. And the fifth one is in a bird. And the only reason that I know that they are not humans is because I know from where they come from. But in fact, histologically, they look exactly like a human mesothelioma. Asbestos, I'm not going to talk today because I do not have the time to cover everything, but we all know that the majority of mesotheliomas are caused by asbestos exposures. In the past, that was the vast majority. Today, this fraction is shrinking. And then there is Karen, Turkey, where 50% of people died of malignant mesothelioma, which makes this the most incredible cancer epidemic in the world, because there is nothing that causes 50% of deaths in a population. Even the infamous Spanish influenza killed only 20% of people who are infected. COVID is killing 1% of people at most, probably less than that. Here, 50% of people die of mesothelioma. The issue is why they die of mesothelioma. The prevalent hypothesis was that when I started studying this, what I learned in books was that there is erionite in the mountains here, in the rocks. People first lived in these caves here, then in the 1960s they built the homes, but they are built with the same material of these caves, and so they were exposed to erionite. Erionite is a fiber shown up here that is identical, I mean, it looks exactly like crossidolite asbestos, and therefore people died of it because of exposure to asbestos. These villages, I don't know if you have seen them. I visited them many, many, many times over the course of years. This is the Koru family, and this is Mrs. Koru. Now the house is abandoned. This is Tuskoi. Very poor houses, very poor homes, usually a 2-bedroom home in place of a garage with a Mercedes. You can see here a donkey. And you can see this fiber type of materials inside these houses. This is the village of Tuskoi, another village with 50% incidence of malignant mesothelioma. And this is my first visit to the village. I went there by chance. Dr. Barish had invited me to give a talk at the Turkish Thoracic Oncology Society. He was the president at that time in November 97. And I read about these villages and I asked Dr. Barish, I'll come to Ankara to give a talk, but you need to take me to the villages. I want to see the villages. So I went there this day. I thought it was a one-day visit and a touristic visit to the village, which instead turned to be a long-time project and research project. Let me show you what I saw. So people first lived in these caves here, and then they moved in 1960 in building these homes here, built with the same material that they cut out of the mountain. Here I'm inside one of these caves. This is the pharmacist of Karen, and I'm pointing here where he used to sleep with the rest of the family. He and all the brothers lived here. Here you can see the heater where they put the brace to keep themselves warm at the night. You can imagine the house is built entirely inside the mountain. There is a lot of aeronite into there. They all bred these fibers, and so they were full with these fibers. They took me to visit what they call the homes of death, the houses of death, where everybody died of mesothelioma. And we found these kids who were playing cowboy in there. And as I walked through the house, I was covered with this dust, covered of aeronite. And this kid came with a broom and cleaned me up with the broom. And I was in this dust of aeronite. And I have to admit, I was scared. But I thought, I cannot tell the kid to stop because she's playing here. They're all covered with it. If I tell her to stop, she's going to get scared, and I don't want that. Or maybe I was scared myself, and I wanted to convince myself that this didn't make any sense. If the people who lived in this house all have died of mesothelioma, why these kids were not going to die of mesothelioma? Everybody should die. Why only 50%? To make things worse, when everybody died in a house, they demolished, this is Tuskoi, they demolished the entire home. Well, now they left all the rubble here and the dust went everywhere. So there was no way that somebody could be more exposed than somebody else. Everybody was. So I started to look at it. And this is how you do science in a situation in which you do not have the resources to do science. So I got a map from the local municipality and I started drawing circles. And what is orange here is homes where people with age below 40 years of old had mesothelioma. And what is purple is where nobody could remember that there was a mesothelioma. And you see these houses are just next to each other. Look at this. Barish tells me that in this house, everybody died of malignant mesothelioma. And in this house, nobody. And I was sitting next to Barish, standing next to Barish, and I say, Dr. Barish, how is that possible? He said, well, because here there is more aeronite than here, and the type of aeronite here is more dangerous than here. And I said, how do you know? He said, well, because everybody dies here and nobody dies there. And I said, I don't believe it. It makes no sense. So that Barish basically told me that I was a fool young kid who didn't understand anything. He was more polite than that, but that's basically what he said. And so the way that we started being Barish was not the right way to make the list. People in the villages were asking me, the American expert, Why are we dying of mesothelioma? I had no idea. I had studied all those years and I had no answer. Actually, I was asking them questions to try to understand what was going on because I had no idea. Then this guy comes with this kid in his arm. His mother, her mother, had died of mesothelioma the week before. And he tells me, what can I do so that she doesn't die? And I felt terrible. I had no idea. I didn't know what to answer. I knew nothing. So I decided I was going to study that. I went back to America and I told my chairman of pathology, who was a very nice man, that I was going to study this because I wanted to help these people. His name was Ken McClatchy. Ken told me that I was crazy, that I was going to ruin my career and that nobody would care in the United States about an epidemic of mesothelioma in 3 villages in the middle of Cappadocia. I said, I don't care that nobody cares. I care, I want to do it. He said, I cannot authorize you to do that because you are a young man and you are full of enthusiasm. I was like that, but you are going to ruin your career. You can't do it. I said, well, I still have vacation. I can go on vacation where I want, right? So I'm going to spend my vacation in the villages. And that's what I did. And so for the first years, I went there for free when I could during my vacation time. We would put a desk. This is Sarrider, the 3rd village where there was this terrible incidence of mesothelioma. And then people would come and talk to us. This is Dr. Barish. This is Dr. Sally Emery, who worked with us for all those years. And then people would come and tell us, well, I mean, not just people with mesothelioma, but people who had problem because the husband drink too much, because they have a wart. I mean, one after the other, the villagers would come and talk to us. And we would talk to all of them. And I was trying to make sense of what was going on. After some time, Dr. Harvey Pass, the director of thoracic surgery at New York University, started to come with us. Here we are in the municipality of Karen, where we are visiting on the people. It's a very different situation than in the United States, where you see only the patient. As you probably know, in these villages, all the family comes. So you have the patient, you have the nephew, who translated to me, she spoke English, you have the son, you have the father-in-law. It's a discussion with the family that you have. Anyway, and we went many, many times to these villages. This is another visit there. You see Dr. Rhodes Polak, Jenko, the Muta of Karen, Dr. Anin Yang that was with us, Dr. Paz. And we concluded that this was a genetic problem. And we concluded that because by put together pedigrees, this is family 1, you have here 17, everything that is black is a mesothelioma. So you have 34 people here, 17 with mesothelioma. Initially, it was very difficult to understand that this was a genetic disease because people who married into the family had mesothelioma. That's against any logic of genetic disease. But then we found that the people who married into the family, this one, for example, is coming from another mesofamily. If you look here, this person here marries into this family, but she comes from another mesofamily. And then you have that when somebody from a mesofamily marries in a non-mesofamily, you start seeing mesothelioma in the descendants. And we realized that, in fact, they were all related. And the reason that we're all related is that people from nearby villages were scared to marry with them. And so there was a lot of intermarriage within the village, even if they didn't say so. But the fact is that within 2 or 3 generations, they married with each other between families because the other villages would marry with them. And so that's what we were seeing here. So we say, okay, there is a genetic predisposition to get mesothelioma. Probably people who are genetically predisposed are those who, when exposed to erionite, develop mesothelioma, gene-environment interaction. I applied for a grant to the National Cancer Institute to isolate this gene, and I got the grant. People told me that I would never get the grant because nobody would care about 3 villages in Turkey. And in fact, I got the grant. Dr. Barish, Dr. Emri, and Dr. Dogan represented the Turkish core, and they worked with us on this project. We published our first paper in Lancet in 2001. Now, as I told you, when I started this discussion with Dr. Barish, he disagreed with me. Actually, he thought really that I was an arrogant kid. And so I continued to work without Dr. Barish to try, and I put together this information that you see here. And then I published it in Lancet in 2001. Before the paper was published, I sent the manuscript to Dr. Barish out of courtesy. I said, Dr. Barish, out of courtesy, I want to let you know that I continue to work with that, with some other of your collaborators that agreed to work with me. And this is what we found. And we have the paper in Press and Lancet. So Barish writes back to me and say, I don't believe you. Send me the pedigree, I will check. So that didn't surprise me. I put together the pedigrees and I sent to him. 2 weeks later, Barish writes me back, says via email. Barish used email. Barish said, I went to the villages. I checked your pedigrees. Except for 2 mistakes, they are correct. So now I believe you and I want to work with you to find the mesothelioma gene. At that point, I realized that I was totally wrong about Dr. Barish. I thought that he was arrogant and that was impossible to talk to him. And this man, who was very famous, was willing to say he was wrong and now he wanted to work with me. I mean, it takes a lot of strength to say that. Very few people will have the strength to do that. So we became great friends and we spent a lot of time together trying to figure it out what was going on. He studied genetics. He wanted me to give him a book of genetics. He studied the book of genetics. We spent hours discussing this thing together. And I think we became greatest friends. I mean, it's one of the person that I admire the most is Dr. Barish. And Dr. Barish really deserves full credit for this project of Pathology. So what do you do when you find that is a genetic problem and you now have to find the gene? We got the grant to find the gene. What do I do? So I went, do you go back to the lab and say bye-bye, or can we do something for them? This is the village of Sarıyer. The village was abandoned in 1959, this part, because you see here, this is the Red River. On the other side, there is the new village. The government built a new village because this old village kept to get flooded. This man here is a good friend. I can't remember his name now. He died this year of malignant mesothelioma. And on the other side of the river, they built the new Sarıyer, the Turkish government built the new Sarıyer with bricks and cement. And at that time, there were no cases reported of mesothelioma. So we thought if we could get new villages for Tuscoy and for Karain, maybe even if people are genetic predisposed to it, they are not going to get mesothelioma if they are not exposed to erionite. And we were talking, I don't know if you know the place, it doesn't exist anymore, but along the Red River, there was a beautiful restaurant on the water, on the Red River, where you would eat trouts. You would go there, they would catch the trout for you, they were growing trouts and cook them for you, and was delicious. And that was the place that Barish preferred. So at night, after we worked in the villages, we would go there and eat the trouts. We were there, it was about 9 o'clock at night. Beautiful day, the stars were out, and we were discussing the situation. And I tell Barish, can't we go and talk to the Turkish Ministry of Health and tell him we need to build new villages to save these people? And Barish said, forget about it. Politicians are useless. They make a lot of promise, they do nothing. I've gone to them a lot of times. I don't want to waste my time. So as we are having this discussion, there is this guy who is serving the trouts, the waiter. And he says, I can't remember if it was his uncle, I think. My uncle worked for the Turkish Ministry of Health. He works for him directly. If you want, I can call my uncle and see if he can get you an appointment. I say, please call. But he said, forget it. Anyway, the guy calls. And the guy answered on the other side and say, okay, my uncle say he can get you an appointment for, it was about 4 or 5 days later. I said, okay, tell him we'll go. So Barish didn't want to come. I said, please, you have to come. Eventually, we convinced Barish to come. I canceled my trip to go back to America. And then we go to meet the Turkish Ministry of Health. We go there and we were completely unprepared. I didn't know. I thought Barish was going to do the talk. I was with Barish. Sally Emery was with me. Umran Dogan was with us too. And we are sitting there waiting to go there. We are called to meet the Turkish Ministry of Health. So we get up. And as I get up, Barish tells me, you talk. Because since you are American, maybe he's going to listen to you. He's not going to listen to me. I'll translate. So I had prepared nothing. And now I have to talk to the Turkish Ministry of Health about it. So I didn't have time to think. I just sat in front of him and I explained the situation. The Turkish Ministry of Health seemed a very nice person, didn't say a word. At the end of my speech, he said yes. That's the only word that he said. And then they told us that we had to move on because other people had to come in. So my impression was that I wasted my entire time in there. I canceled my trip for nothing, and why the heck we had done so? A year later, I go back to Cappadocia. When I go back to Cappadocia a year later, everybody were all smiles, and the governor of the region give us a car, this car here, and they take me to the village of Tuscoli. And they say they had a surprise. And when I arrived in Tuscoli, they had built a new village. I couldn't believe it. I had never found in my life a politician who promised me to do something and actually does it. I had to go all the way to Turkey to find someone who tells me I'll do it and he does it. And I really couldn't believe it. And they built all these homes. This is 1 year later exactly. Now Tuscoli is a total new village, if you have seen it. And I couldn't believe it. And in this car, when they took me there was a journalist. So I told the journalist this story. The journalist put it on the next day. I told the journalist, this guy puts it on the newspaper in Ankara, in the main newspaper in Turkey. There was out there. The Carbone said that he had to come to Turkey to find the first politician in his life who maintained his promise. Well, that made me pretty popular with the Turkish Ministry of Health at that time. And they sent Dr. Murat Tuncer, who was the director of cancer control, to talk to me. I talked to Dr. Murat Tuncer. Dr. Murat Tuncer asked me what else they could do for the villages. And I said, we could build a hospital for them because they have to travel to the nearby villages. They're poor people. They don't have car. If we could have a little clinic for them. And Murat said, that's a great idea. And within a year, and this was built because of Dr. Murat Tuncer, he deserves 100% of the credit. He got a clinic for the people in Tuscoli where they could be seen by the doctor so that they would not have to go far away. Now, I have to say that I've never seen in any part of the world a country that has been more responsive to take care of their people for nothing, because this was done because they care of their own people, not to get votes, not to get anything. So I was mostly impressed by seeing that, and that increased my admiration to Turkey. A year later, we went to open the hospital. Murat Tuncer didn't want to come. He didn't want to get any credit for it, he said. So he sent his number 2, who is here, and his number 3, who are here. I can't remember their name there. This was the first doctor of the village of the Tuscoli hospital. And this is my friend, Mr. Tunç. Mr. Tunç is the tailor of the village of Tuscoli. And he helped me tremendously in putting together those pedigrees and in figuring out that this was a genetic disease. This is Dr. Harvey Pass that came with me. And this is Dr. Sudhir Srivastava that was sent by NCI, the National Cancer Institute in Bethesda, to see this whole thing. So it was a pretty nice thing. And this is Dr. Aning Yang, with whom I ran the lab together in Bethesda and with whom I work. In the meantime, in my lab, we were studying this condition and trying to find the gene. And we were 4 years and 6 months into the project, and we had nothing to show. We had got a grant from the NCI to find the gene, and we couldn't find anything. We were underpowered. We didn't have enough genetic information. It was impossible to find this gene with the amount of material that we had and the amount of work that had to be done. And the story would have ended, because after you get a grant, if you do not publish even one paper after 5 years, nobody will renew it. Was that as I was studying the Turkish family, American family started to call me and say, "You are going all the way to Turkey, Dr. Carbone, but we are having the same problem here. We have many people."
Hocam, I'm sorry for interruption, Mr. Carbone, but there is a little problem about your microphone. Can you just fix it? Because it's really difficult to hear you.
Can you hear me better now?
Yes, thank you.
Okay. Sorry about that. Thank you for letting me know. So there were these families in America who called me and told me that they had the same high incidence of mesothelioma that I was studying in Turkey. So I started to study them. And it was easier to study the American families because in Turkey there is the problem that you cannot bring the DNA to America to study because of complicated laws. But in America, I could get all those DNAs. And so I had two families here, one from Wisconsin and one from Louisiana. The one from Wisconsin has 5 mesothelioma, 1, 2, 3, 4, 5. The one from Louisiana is 7 mesothelioma. This is the sign that tells you that it's a mesothelioma. One of them, Mary, had mesothelioma and eye melanoma. This was the second eye melanoma in this family with mesothelioma. I checked with my biostatistician who told me that the chance of developing uveal melanoma and mesothelioma was less than 1 in 10 million, between 1 in 10 million and 1 in 100 million. I thought that there could not be a chance. And so I put my entire team to study chromosome 3p. That is the chromosome that is most often altered in both mesothelioma and uveal melanoma. In other words, when I look at commonalities between melanoma, eye melanoma, and mesothelioma, genetically the commonality was on 3p. There was a high incidence of mutations in 3p in both tumors. And when we did that, we discovered that all the people who had cancer in this family had a mutation of this gene called Papuan, which at that time had not been linked to human cancer. So we published this paper saying that germline Papuan mutations, heterozygous mutation, meaning only one allele is lost, predisposed to malignant mesothelioma. They could cause mesothelioma by themselves or better upon exposure to asbestos. And in fact, here, this is the first immunohistochemistry. I did it myself. This is a mesothelioma from a wild-type BAP1 person. You see the BAP1 protein staining. And these are from people from these families, all of them showing lack of BAP1 nuclear staining, because the second allele is lost in the tumor, and so you have biallelic inactivation, which is also proof of pathogenicity. These people we find also carry some, most of them, not all of them, carry these lesions that are pretty common. Most of us has something like that, but histologically they are unique, and I don't, you see, they are often close to a melanocytic nevus, for those of you who are pathologists, This is a normal melanocytic nevus. The lesion, however, has these large cells that look like a Spitz lesion, and these are very large cells near the melanocytic cells, and this is an entirely intradermal lesion. And when you do a BAP1 staining, you see that the melanocytic cells have nuclear BAP1 staining. Instead, these lesional cells lose the BAP1 nuclear staining. We call these melanocytic BAP1 associated intradermal tumors. These are benign tumors. However, they are characteristic of people who carry these mutations. And so this is, we discovered with that the BAP1 cancer syndrome that I named in 2012 and then in 2013 in Nature Review Cancer. And people who are born with germline BAP1 mutations develop 29% develop mesothelioma, about the same number, 28% uveal melanoma, 20% cutaneous melanoma, 11% clear cell renal cell carcinoma, 10% basal cell and squamous cell carcinoma, 7% breast cancer, and down here, and then 15% of them MBATs, but 15% is an underestimate because very few people go and check these things because these are benign lesions. I think that if everybody would check, would be close to 100%. But since these things are not checked because they are benign, we have only that 15% of the patients reported today have it. So after we published our paper, there was a lot of skepticism, as always in science. When you say something new, people think you are crazy. But in fact, as of now, there have been over 800 papers that have confirmed the data, and there are 80 papers from PubMed in which we find 574 individuals with 841 cancers. 100% of people who are born with BAP1 mutations develop cancer. And obviously, several of them, 29% of them, have anyway between 2 and 7 different cancer types have developed. So this is a new condition that is considered today an ideal condition to study gene-environment interaction because these individuals who have BAP1 mutations are very susceptible to asbestos and UV light or radiation and develop mesothelioma, uveal melanoma, and other cancers. We demonstrated the susceptibility to asbestos in a mouse model that has an heterozygous mutation of BAP1. When you give to a wild-type mouse asbestos, a low concentration, which is 0.5 milligrams total, only 7% of them or 8% develop cancer. But when you give the same low amount to a mouse that carry an heterozygous BAP1 mutation, you go up to 30% of cancer. When you give the full dose, that is 5 milligrams, you have about 30% of cancer in wild type and about 75% in the mutant mice, showing the interaction between the environmental carcinogen asbestos and the genetic mutation. From where did these mutations, at least in America, come from? We published that in PLOS Genetics some year ago. This is an interesting paper, I think. We found that several families carry the same identical mutation. So the question was, is this a hotspot mutation or are these families related? We had 4 families with this mutation. One thing you may want to look at this family, we will talk later. Look at this. These patients get a leiomyosarcoma age 32, uveal melanoma age 48, peritoneal mesothelioma age 55, pleural mesothelioma age 60, giant cell tumor of the bone age 71. In other words, these people get one cancer after the next, but they don't die. And that was what caught my eyes. Leiomyosarcoma, you're dead in 1 year. Uveal melanoma, you're dead in 1 year. Mesothelioma, you're dead in 1 year. These people keep getting cancer, they don't die. What was going on? Anyway, back to this family, we found that the family originated from Switzerland, went to Germany, and moved to the United States in 1717. And we found 4 of these families initially, and we put together a pedigree of 80,000 people, all the way back to 1524 and 1520 to this original couple in Switzerland. You say, well, why you do that? What is useful for? Well, because when you find somebody with a mutation, you can go up in the pedigree, and then you can go down and identify other family members that you didn't know if you were just here. But this brings you the rest of the family who also have this cancer, and many of these cancer can be prevented by early detection. So you can contact these people who will benefit from screening, and that's what we did. And then we thought, okay, let's go to Switzerland and see if we can find them, the original family, and we did. We found that this is Isabel Opitz, that Muzaffer Metintash knows well, and I think many of you may know her. She's now the chief of thoracic surgery at the University of Zurich. And with her help, we found the original house from which the family immigrated in the 1488 to Germany. These are the direct descendants of them. And then we found a branch of this family with uveal melanoma and malignant mesothelioma, and showing that, in fact, this mutation came to the United States through this family. And now we have 10 different families like that in the United States, including one that includes the former president of the United States, Truman, but he didn't inherit the mutation. So what about sporadic mutation? Is BAP1 mutated besides the germline in mesothelioma? Is BAP1, are sporadic mutations frequent in mesothelioma? We published that 22% of sporadic mesothelioma had BAP1 mutation in our original paper. Bott et al. Found the same thing. And then there was this paper by Yoshikawa to say, no, you're wrong. It is 61%. So I established a collaboration with Dr. Yoshikawa and Dr. Mitsuru Emi, who had said that we were wrong. Of course, I thought we were right and that they were wrong. This is Dr. Masaki Nasu, who did all the work in my lab. And this is Dr. Mitsuru Emi and then Dr. Yoshikawa. And when we did this study, we found out that actually they were right and we were wrong. And that is 60% of all mesotheliomas carry BAP1 mutation. And the easiest way to see if they carry BAP1 mutation is immunostaining. Better than any genetic test, because when BAP1 is wild type, you find nuclear staining. When BAP1 is mutated, either you find nothing, meaning the specimen is totally negative for BAP1, or you have only cytoplasmic stain, because all the mutation block the ability of BAP1 to go in the nucleus, and so you see only nuclear staining. So unless you see this, you know that you have a biallelic inactivation of BAP1 in a mesothelioma. So the question was, why is that we missed it? Why we and Mark Ladani and his team at Harvard missed it, and Dr. Yoshikawa was right. Why we missed it? Well, we missed it because we used NGS and Sanger sequencing. Sanger sequencing has been designed to identify nucleotide level mutation, point mutations. So, for example, in this case, you can identify a specific point mutation in this particular patient. But when you have a large chunk of the DNA that is lost, here you have exon 7 to exon 13 deleted in one of the alleles, you cannot detect it with NGS. NGS is not designed to detect that. Instead, we detected that with MLPA, which is designed, or you can detect it with whole genome sequencing. So NGS is going to miss all these large deletions and underestimate the number of mutations present when the deletion is large. And then sometimes neither MLPA nor Sanger would identify mutation at splicing sites. And the mutation at splicing site we'd only detected when we sequenced the RNA. Then we identified that this mutation at the splicing site has caused this massive deletion. So you see how the different type of technology that you use can give you different results. We studied together with Dr. Yoshikawa how was BAP1 mutated in mesothelioma, what kind of genetic deletions there were. And what we found studying chromosome 3 is that you do not have an entire deletion of the gene like you have, for example, in renal cell carcinoma, where a large chunk of chromosome 3 is deleted. Instead, you have lesions that is like a knife cutting 2,3P. And so you can have specific genes within the genes that you have these deletions, but you do not have the entire piece that is deleted, which made us suggest that what's happening is chromosome shattering, that then they get reassembled together. So how does BAP1 work? Why is BAP1 so important in causing cancer? That was a big question, because there are very few genes. P53 and RB are the only one well, not the only one, but among the very few that will cause cancer in 100% of people who carry the mutation. Most genetic mutations, for example, BRCA1, BRCA2, will cause cancer in 50% of women, but in 100%, why was BAP1 so powerful? And how do you study something when you know nothing about it? When we started studying BAP1, nobody knew anything about it. So it was a fishing expedition to try to figure it out. How did we do that? Well, we published that in this paper that you can read if you want to find the details. But to summarize, what we did was we got volunteers from these families. We got people matched in the same family for sex and age, and we got their cells, we got a skin biopsy, and we put the cells in tissue culture from a wild type and a mutant member, wild type and a mutant member. And you see that those who carry germline mutation have about half of the BAP1 protein. Then we put these cells in tissue culture, and we did all sorts of possible experiments with them to see if we could figure it out, what was difference in these cells compared to these cells. And most experiments failed, as you can expect. But eventually we got lucky and something worked. And to summarize, this is what we found. In a BAP1, in a normal cell that has normal BAP1, if the cell is exposed to asbestos, UV light, ionizing radiation, chemotherapy, anything that causes DNA damage, you have DNA damage, and BAP1 helps repair the DNA damage. If the damage cannot be repaired, BAP1 is a deubiquitylase that stabilizes the channel that transfers calcium from the endoplasmic reticulum through the cytoplasm into the mitochondria. Calcium in the mitochondria is required to start the cell death, apoptosis, and so cells that have accumulated genetic damage suicide so that they do not pass the genetic damage to the daughter cells to prevent cancer. Normally, IP3 is ubiquitinated by this ubiquitin ligase. BAP1 removes ubiquitin, and that maintains the functionality of these channels. Once you have no BAP1, this channel is ubiquitinated, it's degraded. Calcium remains into the endoplasmic reticulum, cannot be released. At the same time, DNA cannot be repaired. So now you have excessive DNA damage, but the cell cannot suicide, and therefore you have accumulation of cells with genetic mutations that grow and eventually give rise to cancer. Moreover, the absence of the reduced amount of calcium caused major metabolic alterations. We found that we could, by doing a metabolomic analysis without any genetic testing, distinguish with 100% reproducibility wild-type individuals from mutant BAP1 wild-type. What was different was that the glycolysis was increased in BAP1 mutant patients. Here you have a BAP1 mutant versus a wild type fibroblast in tissue culture, and you see how glycolysis and glycolytic capacity is much increased in BAP1 mutant cells. You can easily induce that in any type of cells. You can take a wild type BAP1 cells, you silence BAP1, and you increase glycolysis. You can take human mesothelial cells, you silence BAP1, and you increase glycolysis. Vice versa, you can take a BAP1 mutant fibroblast, which is this one. You put back wild type BAP1 and you decrease glycolysis. And if you put a mutant BAP1, you cannot decrease glycolysis. When you measure the mitochondrial respiration into these cells, you see that all the parameters that all the parameters that measure mitochondrial respiration are decreased in BAP1 mutant cells. So these cells are not deriving energy through the Krebs cycles, but through glycolysis. And again, you can do the same thing. You can take a wild-type cell, you can silence BAP1, and once you do that, you reduce all the parameters of mitochondrial respiration. You can do that in mesothelial cells, and vice versa. You can reestablish mitochondrial respiration by putting back wild-type BAP1 into BAP1 mutant cells. So BAP1 mutant cells produce most of their energy via aerobic glycolysis, and that's which is called the Warburg effect. But usually we think of the Warburg effect as something that associates with cancer cell. Here you have a Warburg effect in a normal cell. Why is this important? Because the Warburg effect helps cells grow and invade nearby tissues when there is lack of oxygen around. So because these cells are already in a glycolysis metabolic situation, once there is accumulation of genetic mutations, these cells are already ready to grow and invade nearby tissues, which explains why they are so oncogenic. So for those of you who are clinicians, who cares about all this biochemistry? Well, the reason that you care is that, as I mentioned before, as studying these patients, we published this first in 2015, we found that they don't die compared to the mesothelioma patients. Their survival is much, much better compared to the mesothelioma patients. Why is that? For those of you who are pathologists, I'm a pathologist, this is how these tumors look. This is a trabecular epithelial type mesothelioma. You see all parallel things. If you look at it, it looks malignant. But if you go at low magnification, you see that there is no invasion. This is all intrapleural growth. Layers and layers, but this is all intrapleural growth. There is no invasion. You see that? This looks very benign. On the other hand, it doesn't look benign because there is too much of it. Really too much, but there is no invasion. This is all intrapleural growth. So if you want to be strict, I mean, this is obviously mesothelial cells. In case you have doubts, this is a calretinine stain. This is a calretinine stain. You see the normal cells here? And this is, they're going in, but they're not going in. This is all pleural. They're not invading anything. There is no fat. There is no muscle. This is all pleural. You see that? This is WT1. Very nicely lined it up. These are the normal cells. These are the cells that keep growing. So since there is no invasion, if a pathologist were to call it atypical mesothelial hyperplasia, you couldn't say he is wrong. But my experience tells me that this is malignant. And the reason is that these people get cancer. You see here that they lost BAP1 staining with biallelic inactivation of BAP1 in its evidence of malignancy. You see that? There is no nuclear staining. There is still some in the mesothelial cells normal, the lay here, but in the one that proliferate inside, there is total loss of BAP1, complete loss of BAP1 in these cells. So this lesions eventually are malignant, but they progress very slowly, and that explains why these people live so long. And that influences, of course, how you want to treat them. And we are going to have a discussion on that on February 13. You are all welcome to participate if you want, to discuss what is the best way to treat this patient given their prognosis. So when we say that people with Papuan mutation lived longer, again, there was a lot of skepticism about it. Two groups eventually studied this. One was at the University of Chicago with Dr. Churpek, one by Dr. Hassan at the National Cancer Institute, and we studied that in more detail. And fortunately, all of us agreed, and that is that the people who have mutation of Papuan or other germline mutation live longer. So in the second study that I did, based on my experience, I hypothesized that the family history of mesothelioma, personal or family history of multiple malignancy, or patients younger than 50 would help us identify those meso linked to germline mutation of Papuan or other genes. And so over the course of 7 years, we enrolled mesothelioma patients with this characteristic. The rate male to female and pleural to peritoneum was 1:1, which immediately tells you that this is a unique group of people. We found that over 50% of these mesotheliomas occur in carriers of pathogenic germline mutation. 50% of them are Papuan mutation, and they survive much longer. About half of these patients carry Papuan mutations, the others carry mutations of other tumor suppressor genes. This group of patients rarely report asbestos exposure. This is the survival of the patients that we studied, compared to overall survival in mesothelioma in green and survival in stage 1 mesothelioma. You see that we have an average survival of 6 years compared to 1 year. So these people do significantly better. And you know that you are dealing with the mesothelioma patients which fits into the germline mutation when he or she has a personal or family history of multiple malignancy, when the patient is younger than 50, or has cancer linked and they have mutations of Papuan or other genes. The study by Churpek at University of Chicago was on 198 mesothelioma patients who were not chosen for age like we did. They just got them all. And they found that among all mesotheliomas, 12% of them carry germline mutation, and 20% were in those of 55 years or younger, compared to only 4% in people who are older than 75. So you see, young age indicates a patient who carried a germline mutations. They were rarely exposed to asbestos. In the study at the NCI with Marie-Claire King and Dr. Hassan, 293 patients. Again, 20% of all patients not selected carried germline mutation, and they had a median survival of 7.9 years. In addition, although Papuan was the most common mutation in all studies, in the study at the University of Chicago and in the study at the NCI, also other germline mutations that are known to cause other cancer syndrome were reported. For example, BRCA2, MLH1, TP53, etc. So mesothelioma can occur also in this other well-known cancer syndrome, although it's not that frequent, and that's why it was not reported previously. But you see that all the study found that people with the Li-Fraumeni syndrome, with the TP53 mutation, can also develop malignant mesothelioma. In addition, people with Papuan mutation, some of them have mutation of other tumor suppressor genes, like MLH1, which cause other well-known cancer syndromes. And so here you have a situation in which you have gene per gene interaction, meaning two mutations, two cancer predisposing mutations interacting with each other to cause cancer. To summarize, there is a subset of mesothelioma with improved survival occurring in carriers of Papuan and other germline mutations, which rarely report asbestos exposure. all mesothelioma patients should be genotyped because this can be beneficial to those who may carry actionable mutations into family members. One of these mutations is BLM, and we did this study together with the research team of Dr. McIntosh. We discovered that, and with other collaborators, we discovered that BLM gene mutations increase susceptibility to asbestos and mesothelioma, and our paper was published in PNAS this past December. Briefly, BLM is a gene that regulates DNA repair, DNA recombination, and DNA replication. It is well known that B-allelic mutations of the BLM gene cause the Bloom syndrome. The Bloom syndrome is characterized, besides short stature and other things, by a very high incidence of cancer, which are reported here. The question is, would heterozygous germline mutation also predispose to cancer? There are a number of suggestions in the literature, but nobody has been able to demonstrate that, and we did so in our paper. We had two families that we studied. The first one is a Turkish family from Dr. McIntosh, in which two individuals had pleural mesothelioma, and they had biallelic inactivation on BLM. They had a germline mutation of BLM and biallelic inactivation on BLM in their tumor cells, as shown by the absence of nuclear staining. The other family is an American family where you have also two cases of mesothelioma in a family that also has melanoma, that is a cancer that is associated with these cancer syndromes as seen in the Papuan. So was BLM causing it? Well, we extended our studies to the team of Dr. Hassan at the National Cancer Institute, and among about 130 patients of which he had germline DNA, 5 of them carried pathogenic germline mutation of the BLM gene with a very CAD score. The CAD score identifies genetic mutations that are pathogenic, that you can identify as pathogenic. To be sure that we were correct, we studied whether a BLM heterozygous mutation would increase DNA damage, and indeed they do. Here you have mouse cells that we got from a mouse that is heterozygous for BLM, like the patients that we were studying. And when we measure micronuclei in these cells, which accumulate in response of DNA damage, you see that there is an increase of the micronuclei in the cells that have BLM mutations once exposed to asbestos or even spontaneously compared to the wild-type cell. The quantification is here. Although they develop genetic damage, these cells don't die. When they're exposed to asbestos, they don't die, similar to what we had seen with the Papuan. And in fact, here we are measuring caspase 3, which indicates apoptosis. And you see that once you silence BLM, the cells exposed to asbestos do not die. However, these cells, when they are exposed, these cells, when BLM is silenced, secrete high amounts of tumor necrosis factor alpha. Asbestos causes cancer by causing a chronic inflammatory reaction that leads to the release of HNGB1 that then induces TNF-alpha secretion that stimulates the growth of malignant mesothelioma cells. So what we found is that the reduced amount of BLM caused an increased release of TNF-alpha. Indeed, when we injected asbestos in mice, crocidolite asbestos in mice, wild type or BLM mutant mice, we have that in the BLM mutant mice, we had an increase of M1 macrophages that are the tumorigenic, protumorigenic macrophages, accompanied by a major increase of TNF-alpha and interleukin-1 beta, which are the mediators of asbestos-mediated inflammation. So basically, the mutation of BLM was favoring the release of those cytokines that we know cause mesothelioma. And indeed, when we injected wild-type mice or BLM heterozygous mice with asbestos, we had a much higher increase of mesothelioma in BLM mutant mice compared to wild-type mice, proving that the BLM mutation increased susceptibility to asbestos and to mesothelioma. So to conclude, mesothelioma patients with germline, Papuan, BLM, and other mutations are at high risk of developing more cancer and benefit from screening and early detection, which is often curative. Many of these tumors, eye melanoma, skin melanoma, can be completely treated, cured, excised when detected early and then incurable when they metastasize. Family members have a 50-50 chance to inherit the same mutation. They should be tested, as they will also benefit from screening and early detection. Mutation carriers are susceptible to radiation-induced cancer. They should be screened with MRI or with sonogram, ultrasound, not with CT and X-rays that can cause cancer by themselves, especially in these people. Who are the patients likely to carry germline mutations among mesothelioma patients? Those younger than 55. The younger the patient, the more likely is that he or she carries a germline mutation. Women with peritoneal mesothelioma, almost all of them are young women with genetic mutations. Patients with relatives with mesothelioma or eye melanoma or renal cell carcinoma. Once you have somebody with meso who also has a relative with eye melanoma or renal cell, you know that is a Papuan patient. Patients with a strong family history of any type of cancer. Mesothelioma can occur in people with Li-Fraumeni syndrome, with any type of cancer syndrome. Let me finish with Cappadocia, the last 2 minutes. So did I forget about Cappadocia? I did not forget about Cappadocia, but things changed. And so it became more difficult for me to go over there. Then there was the war with Iraq, was kind of complicated. But then in 2018, July 2018, my friend Tunç, the tailor who helped me to do all the studies, developed mesothelioma. And he said he wanted to see me before he died. So I flew to Cappadocia. Dr. Emry picked me up at the airport and we went to the villages. And this is my visit to Mr. Tunç. I want to show you how science can help change the life of people. Of course, the merit goes entirely to the government Turkey, because it was the Turkish government who did all it, but they did it because of our science. So this is the house of Mr. Tunç when I met him. This is the kitchen of Mr. Tunç when I met him. This is the roof of his house. This is the sink of his kitchen. He was a rich man because he was a tailor, so he had a bathtub in the house, and this is his bedroom. This is his house today. This was his house. He died, unfortunately. This was his house today in New Tuskor. This was my office. This was my office now in the nice little hospital that the Turkish government built for us. And they have now 2 doctors working there. At least that was in 1998. They even have an ambulance. This is my friend, Ibrahim Chan. Me and him working in Karain some years ago. This is me and Ibrahim in front of his house in Karain. This was his house in Karain. This is his house today. It even had a dishwasher. So you see how science can make a big difference when there is a government who is willing to listen and to do something about it. And this is my friend Barish, who died, but his dream was to give a new village to these people, and we did it. And I think that he died happy. If you want to read more about it, we published 2 recent papers, one in Nature Review Cancer and one in Cancer Discovery. The one in Nature Review Cancer is on gene-environment interaction. The one on Cancer Discovery is on the Papuan cancer syndrome and mesothelioma. Importantly, look at this one. We have Nobel Prizes and a lot of members of the National Academy of Science on it to show how mesothelioma has now become something of interest outside the niche of people who studied it, but we have the top scientists of the world listed here, who are all interested in the study of gene-environment interaction and that recognize that mesothelioma is the perfect cancer to study. This is my lab in Hawaii. This is Harvey Bassi. He's not in our lab, but I've been working with him for 20 plus years. And so that's why I put the picture here. And this is Dr. Aining Yang, with whom I run the lab together. And this is my team in Hawaii. And thank you very much for listening. And if you have any questions, I will be glad to answer. Thank you.
Thank you so much, Dr. Carbone. We have some questions from audience. They thank you so much. Dr. Serdar Altınay ask you, what do you think about mesothelioma in situ? Can be lack of BAP1 expression a sign of mesothelioma in situ?
So the old definition of mesothelioma in situ was that you could call mesothelioma in situ a lesion only if you had another area where the mesothelioma was invading. And so you could go to the other place, say, okay, this is in situ. I think that the question is correct. If you see a lesion that you do not see that is invading, but you have absence of BAP1 immunostaining or P16 immunostaining, then most likely, you can call that malignant mesothelioma. It's an early stage, but you know that you are dealing with a cell that is malignant. If you have a lesion that is hyperplastic, hyperplasia, atypical hyperplasia, and you have a wild-type BAP1, I would call that atypical hyperplasia. You must have at least either P16 or BAP1 mutated. If you don't have either one of them, that's benign.
Okay, he has a second question here about cell cycles genes. Are there any relation of CDKN2A for mesothelioma? So can CDKN2A inhibitors be effective for treatment of mesothelioma?
So CDKN2A codes for P16 and P14. So I kind of already answered that. Mesothelioma usually have either mutation of BAP1 or mutation of P16. So if you're looking at a mesothelial hyperplasia and you stain for P16 and it's normal, you stain for BAP1 and it's normal, that's benign. If one of the two is mutated, that's malignant.
Okay. Binur Hocam, birkaç soru daha var ama şurada görebiliyorum ben. Siz devam edin.
Hocamızın var.
Evet.
I also have a question if you...
Yes. Akif hocamızın mı sesiydi o acaba?
Akif Hoca'nın bir sorusu var. Sorursanız.
Evet.
Okuyabiliriz ya da kendisi mi sormak ister acaba Akif Hoca?
Hocam size göndermiş. Siz iletin artık soruyu.
Peki, ben okuyorum. Dr. Carbone, thank you very much for this eye-opening lecture. And the question is, which type of protein is expressed in cells with BAP1 mutation? Can it be detected by immunohistochemistry? It's asked by a thoracic surgeon. Yes.
You can do an immunohistochemistry for BAP1. If BAP1 is biallelically inactivated, meaning both alleles are inactivated, you are not going to see BAP1 staining in the nucleus. You may still see cytoplasmic stain because a mutated protein may accumulate in the cytoplasm. But if you do not see nuclear staining, you know that the gene BAP1 is mutated.
Okay.
Yes. May I ask one question? You always emphasize in your articles to reduce the percentage, the risk of misdiagnosis of mesothelioma, which delays causes delays in treatment. And to be able to reduce the misdiagnosis of mesothelioma, of course, the multidisciplinary approach, like early diagnosis of early effusions, BAP1, including BAP1 immunocytochemistry, P16 FISH, and what do you think of analysis of biomarker, soluble biomarkers? Because our Swedish colleagues are extensively applying hyaluronan, and some others are applying mesothelin and osteopontin as a know-how package. Would you recommend also?
My friend Harvey Pass is spending a lot of time in trying to identify biomarkers for mesothelioma. The best one that we have right now, I think, is fibulin, that seems to work. But none of them has a level of accuracy at this time that can be used for diagnosis. They are experimental things that we are trying to figure it out how to use. To use, for example, to identify a recurrence. But at this point, you cannot make a diagnosis based on SMRP or any other biomarker.
And what do you recommend about MTAP, methylthioadenosine phosphorylase?
What do I recommend? Means what? I am not sure I understand.
Some publications, mention methylthioadenosine phosphorylase, MTAP.
Yes, I mean, we have to distinguish what is clinical practice and what is experimental. So clinical practice, forget about it. Experimental research, sure, let's investigate it. Clinical practice, no. Clinical practice, you want to make a diagnosis on something that you're sure of what it is. So mesothelioma is something that is going to be calredinin positive, WT1 positive, TTF1 negative, and so on. BAP1 helps when you do not know whether that hyperplasia is hyperplasia in situ, is malignant or not. When you are dealing with that, BAP1 helps because it helps a lot to decide whether, for example, in cytology, helps a lot. Are these cells that I'm looking in a pleural fluid benign or malignant? If you see mesothelial cells that are BAP1 mutant, well, those are malignant.
Okay, thank you. I have a question for you, Dr. Carbone. Is there any effective targeted therapy for germline BAP1 mutation to prevent mesothelioma cancer?
No, there is no. There has been a trial on AZH2 that failed completely, that was done in Memorial Sloan Kettering. That failed, didn't work. And then there are a lot of attempts to target it, but so far there is nothing.
I also have a question. If you thank you for your excellent talk, Mr. Carbone. It is nice to hear from you again. As you mentioned, both BAP1 and BLM mutations lead